Hello, everyone. I hope you're hearing me. Welcome to SPOT, the Science Public Online Talk Series brought to you by the Faculty of Science at the University of Manitoba. My name is Samar Safi Harb. I'm the faculty's lead for Equity, Diversity, and Community. On behalf of the faculty, I'd like to welcome everyone, our students, faculty, alumni, friends, and external viewers today. I hope everyone is doing well and in good health and spirit. The aim of the SPOT series is to support our students and to use science research as a springboard to bring this community together in these difficult times and as we adapt to this new environment of virtual learning and communication. SPOT will feature diverse and engaging speakers from both our science faculty as well as alumni every Friday between today and the end of the summer. So first, I'd like to give a big thanks to the SPOT team, shown here, looking like they're having fun planning this, working behind the scenes for helping bring this to light on a very short time scale. Big thanks also to all of our speakers for their kindness and generosity in providing their precious time that made this series possible. Before we start, I'd like to go over some housekeeping items. These will be very brief talks, 15 to 20 minutes long, followed by up to 15 minutes of questions and answer period. At the end of the talk, you can type your questions in the chat window, which you can find at the top right corner of the screen. The questions will be visible to the audience. I'll be reading these questions out to the speaker. If you encounter any technical difficulties, please send an email to Andrew, whose email is shown at the bottom of the slide. This is being recorded. Throughout the session, videos and microphones are to be kept turned off or muted. In this online setting, we kindly ask you to be respectful, inclusive, accommodating, and understanding of any potential technical issues. We also ask you to not disrupt or display any disrespectful behavior. The moderator reserves the right to ask anyone to leave or to deny access to a subsequent webinar. Now, moving to the fun part, it's my pleasure to introduce our very first speaker of the series, Dr. Jorg Stetefeld, whose talk is entitled, How to Employ Artificial Intelligence to Fight Cancer. Stetefeld is a professor in the Department of Chemistry at the University of Manitoba and a Tier 1 Canada Research Chair in Structural Biology and Biophysics. His research seeks to understand in detail the structure-function relationship of proteins as dynamic systems, how they're involved in signaling complexes related to human disease, and how they can be used for biomedical applications. His research also has laid the foundation for a new avenue of research, designing proteins to help industry, such as oil and gas companies. Thank you, Jörg, for agreeing to be our very first speaker in this exciting journey. So the screen is all yours now.
Okie dokie, I try to. Okie dokie, can you see the screen with the PowerPoint? Can you see me? Can you hear me?
Yes, we do.
Okay, first of all, yeah, thank you so much. Nevertheless, strange feeling not to see anyone. Yeah, especially Samar, Vicky, and Andrew who did a fantastic job to guide me through Blue Jeans. And as you can see already, the title is very tight and dense, and please forgive me. In this 20 minutes, I cannot touch or go into details in great depth. However, you know, I will do my best, and I will start to tell you a little bit about how we see cancer. Then we dive into what's for us a hybrid method approach, and I show you how we develop drug targets, and then I will touch this artificial intelligence approach. Nevertheless, if you have more questions, there's a fantastic homepage. You can read and try to understand a little bit more. And you're, of course, welcome to give me a shout and talk to me about it. Okay, how we see cancer. It's a bit, many of us are affected by it, or members of the families. And when we were approached by companies and to start to think about, we came across a molecule which is called natrin. Natrin is from Sanskrit, which is technically the most important axon guidance cue we have in the human brain. It's the molecule which defines all our chemoattractive and repulsive decisions we make. However, in 80% of all known human cancers, natrin is upregulated. And this makes it for us an interesting system to work with, especially if you take into account the following so-called dependence receptor hypothesis, namely the receptors natrin as a ligand molecule is talking to are dependence receptors. There is a handful of these guys known. Most prominent are deleted in colorectal cancer, neogenin, or ANK5. And the name implies if natrin binds to them, as you see here, the left part of this cartoon, these receptors multimerize, and then these cells survive and they move somewhere or they adhere or these kind of things. However, if you are able to interrupt this interaction or block this interaction, these cells undergo apoptosis, nothing else than a programmed cell death. Ergo, working on natrin and its receptor complexes is for us a very, very interesting system to tackle these kind of disease. Okay, it starts with defining targets, and I worked for several years for Hoffmann La Roche, and it's usually there most of the guys make a wrong decision in choosing the wrong target. And these natrin targets here is highlighted. This is a complex or a super complex. We work with natrin and its dependence receptors. Let's call them, for the sake of the talk, 1 and 2. They are transient. The job of natrin is not to bind them permanently. No, it goes in and out like a mailman, from the Canada Post, delivering its message. And this makes it for us very, very, very hard to work with. Okay. Here's some echo, but I hope you can hear me. The work we do in my group is we combine many things. I will briefly guide you through this kind of cartoon here. It firstly starts with the fact that we have to make these proteins and these complexes. These are recombinant expression systems which are very, very difficult. We use insect cells or HEC293 cells. In a second step, we have to form these complexes. And as I mentioned before, it's not that complicated to make receptor 1 or receptor 2, but then to force them in some kind of interaction we can study this is a huge challenge. Still, the big thing in my group, I'm by training a protein crystallographer. We crystallize these systems, but there the next problem arises. A protein crystal or a natrin 1 crystal has more natrin molecules than we have in the human brain. It means it's a super up concentration of molecules which have to arrange in a proper three-dimensional space, and this is very complicated. So we decided to back up this kind of, and to avoid the overinterpretation of crystal contacts, we do also things with a technique called SAX, which stands for small angle X-ray scattering. We did this in the old days here in the house in Parker, but these days we go there to synchrotron, and these techniques allow us to gain three-dimensional information in solution. And all these findings are then backed up with lots of techniques where we study the interaction domains by ELISAs, or we perform surface plasmon resonance techniques to figure out, is netrin first binding the green guy or the red guy. Then we look for caspase assays because we have an interest to cause cell death, and caspase 3 in particular is a hallmark caspase in apoptosis. And then, of course, we always go back the cycle. Here you see axon guidance assays where we look, come on, these and these modifications of netrin, are they still able to perform their technique? Why we do so, it has simply something to do with the fact that we really want to tackle and feed systems in the artificial intelligence. And here there are a couple of images of receptor netrin complexes, and you see already here, these systems can be incredibly complicated. It's not always that there one netrin binds to one of the receptors. All sorts of molar ratios exist. And the problem for us is that each and every of these has a complete different pharmacokinetic window and has to be treated differently if you go for a drug design protocol. However, to summarize the finding in this kind of integrated modeling approach, we, in my group, we really generate as much as possible information to really dive into the intimities of these complexes by combining three-dimensional techniques with lots of biophysics or techniques to label things properly. At the end of the day, to really generate a perfect mapping for our approach. So far, the classical approaches we followed, but this has something to do with the funding culture in Canada and the environment we are in now. In the past, we did lots of things with antibodies. It's still the golden standard in the field. And here you see a complex of an antibody we did. And the antibody is in clinical trials in the European Union to treat people with patients with breast cancer. And you see the antibodies, it's a pretty straightforward. You have your library setups. The advantage is you can go for ligands, receptors to disrupt interaction, but you also can stabilize potential complexes. A classical cavity-based approach is still the good old fragment-based drug design, something I did 20 years ago as a postdoc in Basel, in Roche. And here you see a cavity in one of our molecules of interest, which is, of course, a perfect target to look for small molecule binding pockets and to study them. A little bit more advanced approach, and we do this in collaboration with guys in Austria, is the scavenger concept, which means based on the structure information we generate, for example, we know that this little domain of netrin is interacting with a receptor domain. We design them on a peptide library scan basis, and then we use these, and here you see a model. This is a receptor element which binds this, so to say, miniature version of the ligand, ergo affecting downstream signaling. What we now envision with the artificial intelligence, and this is in collaboration with a company in San Diego in the United States called Atomwise, is really to brute force the power of these neural networks in this kind of approach. What you see here looks a little bit like a boring scheme, but the power of this kind of a scheme is they scan with our data about 10 million compounds within a couple of 5 to 6, 7 minutes, and allow us then to screen an enormous amount of drug targets. The artificial intelligence approach to visualize this is a little bit, I mean, we could talk here for years about this approach. It's a little bit seen as the new kid on the block, though I have to confess artificial intelligence is known or is performed or is studied since the 60s of the last century. But we have an interest in this respect in the so-called cut down the search tree approach. Technically, we look, come on, we want to know where sits the cat, by avoiding an overestimate of the parameters of ratio scale factors. We do this simply by an immense amount of data mining. So to say 10 million compounds at this point we feed the system with, which is an enormous amount of information which has to be mined and analyzed properly. At the end of the day, however, it's a learning agent problem to solve. We permanently have to feed the system with our results to allow to optimize it for further screens. And at the end of the day, it's the nearest neighbor method. It means these two kind of things are in a way going into as much as possible to do. Here we minimize the problem in the most intelligent way we can think of. How we do this in a practical approach is very, very easy to say. We are the guys, so to say, in my lab, we generate the immense amount of information these guys require. People or researchers in the company Atomwise, they perform their screening protocols, and we are then at the end of the process again the people who analyze their findings with our techniques. And this is how it looks like. This is a success story we did with them. Here is an epitope of a domain of Natron 1. It's an electrostatic representation of van der Waals surface areas, and you see these R's is an abbreviation for one of the biogenic amino acids, arginine, and these arrows show, come on, here we think something is going on. People known in the field or know the field see, come on, this is an extremely basic or positive charge kind of area. What we do, first of all, we look for the conformational landscape, and something interesting, however not that new, that for example residues, this is an energy landscape of a residue which is, so to say, an antenna, which is very flexible before the ligand binds, but very rigid if it's bound, showing us that there is between recognition and binding. These are two different thermodynamic events, which are very important to know if you design a drug. The second approach is that many of the interactions we are interested in, beside the fact that they are transient, they are driven by co-receptors. We now have one project going on with them in the COVID umbrella, and there these spike proteins require very, very efficient kind of co-receptor element. Without them, they couldn't bind our surface receptors. And our approach allows us not only to determine electrostatics or Lennard-Jones or distance matrices, it really allows us to translate van der Waals or electrostatics or distance constraints into Boltzmann in delta G values. How this looks like at the end of the day is a target validation protocol, which then these guys with their computer power, and the computer power is immense, as mentioned, 10 million compounds. It's less than a cup of coffee time they spend. And what we do here back in Winnipeg, we have an army of setups in the lab, which technically can be summarized as biosensor tools. To the left, you see a machine called a nanotemper, which allows us to perform microscale thermophoresis experiments in high throughput. And to the right here, you see an octet setup for determining biolayer interferometry values, which are two parameters we require to understand the interactions. Okay, what's the future? The future is a little bit here shown to the left. You see how Erwin Hübner started in the 70s, the electron microscopy business here at the university. And to the right, you see a nice picture, blue sky, like today in Winnipeg, and the entry door to MIM, Materials Institute, Manitoba Institute for Material Science, and we, with great help and immense support by the Faculty of Science, are very, very fortunate and very proud that we could afford to get us a cryo 3D TEM setup, which is right now in the process of installment. And this is another kind of a tool, at least in my field, in the structural biology. You know, everyone now goes to high throughput studies of protein and these protein complexes with cryo-electron microscopy. And what you can do, and this is stuff we did in collaboration with people in Europe so far. Here you see, for example, this netrin molecule as an extended rod, so to speak, and here this little circle is the FAB fragment I showed you before, which is right now used in Lyon at the French Breast Cancer Research Council to treat patients with triple negative breast cancer. But also there in the future, you know, things we did here in these low-res negative stain images, you can study at atomic resolution how netrin forms its netrin dependence receptor complexes in solution at more or less atomic resolution without the big problem to get crystals. I forgot to mention this, and I always avoid to say this, you know, if we have grad students, it can be very frustrating. The success rates in these crystallization projects is very low. Okay, coming to the end, lots of people to thank for. Of course, our collaborators all over the world. We are very proud. We were the first Canadians who got an Ames Award from AtomWise. Now, 2 weeks ago, we got our fourth, which always means, you know, they do lots of computer power on our behalf, and they, because we became a partner lab, they usually synthesize between 70 to 100 of their targets, and we can test them as physical compounds. I'm very thankful to our funding support, you know, CIHR, CFI, and I am now in my first term after the tier 2 chair. I'm now a tier 1 chair, and it's great to have this kind of package. And of course, my great friend and companion in co-grad, Dr. Greg Thomey. And here are the synchrotrons. We travel all over the world, not so much here anymore to Villach in the Schweiz, in Switzerland, but the guys in Chicago, in England, and in Saskatoon. Yeah, and what would I be without them? I have the great honor, and I feel very privileged to work with these fantastic people, the members of my research lab. They go for the fire for each other, and yeah, it's very hard for them. As we all know, work is hard, but no work is hell. I'm looking forward to see them in 3D again, and yeah. There are lots of fun ahead of us. Thank you so much for your attention.
Thank you very much, Jörg, for this nice talk. So we're gonna start the question period. I guess you could start writing your questions in the chat window, and we'll take it from there. I guess Jörg, you could read the question if you like.
No, where is it? The chat
the
thing.
Chat button. So there is a question from Dan Martin. Does netrin appear to be upregulated in cancer?
Yes, it's an autocrine, and something it's not known how it goes. But cancer cells obviously are hitchhiking netrin-1 and use the immense power. You know, netrin-1 is, for example, became very famous in the mid-90s when it was found, you know, in multiple sclerosis. Again, I should start with netrin was originally described as an axon guidance cue. Technically, people found it in the brain. And there the job of natrin is really to move cells according a concentration gradient. It's a morphogen. And it had first kind of importance when people understood or started to get a glimpse on multiple sclerosis or the increased amount of damages at the myelin sheath, because natrin at this point is the molecule responsible for guiding the oligodendrocyte precursor cells, which are the repair machines for these myelin sheath damages, to the locus of action. Of damage, sorry, and there it obviously there are a couple of problems. Why cancer cells or how they do so is not known, honestly, or the mechanism of upregulation is not understood. However, the point of natrin is, in this particular case, it's very, very bad. If natrin is upregulated, it's usually a sign of huge problems. Why? Beside the fact that natrin can move cancer cells extremely efficiently from A to B, it has also the very, very dangerous, a companion is the property to melt down the epithelial barrier, which is usually a protection shield to our cells are surrounded by these things which we call the extracellular matrix, which is technically the supply system for food, for nutrients, for signals, for calcium, for water, you name it, for glucose. And on the other hand, it's also an environment which keeps our cells, so to say, in a nice, cozy environment, and it avoids that our cells melt to each other. And natrin is not only moving these cells, most likely it's hitchhiked, but natrin also can melt down the epithelial barrier, which is a very bad thing, you know. I don't know if this answered your question, and I can't see your face, but this is what I would say.
Okay, thank you. So there are a lot of follow-up questions on that. So the next question is, can your biosensors follow an allosteric change due to the binding to secondary site on a protein? From Albert Friesen.
Yes.
Okay, that was a quick answer.
Yeah.
All right.
Okay, now I can read it. There is a second. Is it okay, Samar?
Yes, go ahead.
If I read it, or
yes, please go ahead.
Dean Martin says it is interesting. Okay, this is a statement. Mr. or I don't know, I hope it's okay if I say the name, Peter Moreau asked, what happens if natrin is removed during cancer? You are right. You put in the bracket, we couldn't remove it. We follow the following approach. Technically, remove means we build mimicry versions of natrin. This is this scavenger concept, you know. If you know a certain piece, and these are usually 10, 15 amino acid residues only, which are responsible to bind to a certain receptor element, and you know the structure of these 10, 15 residues, we use peptide library screens to build these little 10, 15 residues in form of a, let's put it this way, miniature natrin. And this now is a guy technically looks like natrin and does the function, but it's not natrin. So to say, we hope that this miniature version of natrin binds, and it works, binds to the receptor and ergo blocks the real natrin to bind at the same place, so to say. It's a cooperativity problem. The hope, of course, is, and this is now the tricky part, that you hope that this mini version of natrin binds, but doesn't cause a downstream signaling. It means it doesn't cause the cell survive. You hope that technically the scavenger binds, blocks real natrin to bind in a second, so to speak, and then the cells die. This is ongoing research. Okay, I hope it's okay. Again, please send me an email. I don't know if you like these answers. So, it's Jason asked a multi-part jumble question. What are the computer processing power capacity requirements for doing these analysis? First of all, we have no atom-wise and all these biotech-oriented AI companies. It's they are, let's put it this way, not really black box, but they wouldn't tell us their intimities about the algorithm. It's more or less these kind of cartoons I showed you, and it's more or less, why you can say, hey, why don't you stay with your antibody stuff, which it worked. First of all, we have to face the Canadian funding environment, and we this is stuff we couldn't afford, and we don't have partners for this here in Canada. We worked a lot with Mike Butler and MapNet because llama antibodies are very interesting for us, and because they pass the blood-brain barrier and so on. But this is past. But I didn't go so much into the intimities, but the problem with the systems we work, I don't know if you got the feeling, we work with protein-protein interaction networks. I showed you this little cartoon. The blue guy talks to the green guy, talks to the red guy. Again, they are transient, but they are very, very important. And the problem is the blue guy is the mailman from the Canada Post, and he is only there for a couple of femtoseconds or nanoseconds, and ergo he delivers the mail to you in the morning or the free press, and then he leaves again. The job of Netrin is not to bind these guys forever. It's too busy. It has to do another job. Ergo, we work with, first of all, this kind of transient complex, and the other big problem with all the other techniques cannot handle, we have monstrous, really monstrous interaction interfaces. Sometimes a couple of thousands of square angstroms, and this is stuff which artificial intelligence is perfectly suited to handle. Compute Canada, Westgood, we are members of them and we use them big time. Yeah, I agree. It's a fantastic resource. We are very, very thankful. Are there any distributed at home computing projects currently running or in development? We took part at the COVID when COVID was hot, or is still in for people like me of huge importance. Though funding is hard to get, so we took part of Foldit. It's FOLDIT. You can send me an email. I'll give you the link. It's Dave Baker at the University of Washington in the United States, and they do this crowd-sourced funding approach for clustering PC CPUs to dock things and do things. And these are publicly available databases. You could log in and do things. There's lots of stuff out there, and we did this in the past, and we worked with high schools here in Winnipeg, Fort Richmond Collegiate, to do these kind of projects with young kids and stuff. Yeah. Okay, Dean Martin said, thank you, I appreciate this. Then comes John Yiva. From which disciplines do researchers come to share in your work? You have seen, it's a bit a cultural thing. In Europe or in where I come from Switzerland, you never get a single man grant. The funding is tight like here. But we were forced in the past to collaborate, to work together. The Swiss guys argue, your crystal structures are wonderful, but who cares? You don't cure a single disease. You cure a disease if you work with someone in a clinic, and if you work with someone in the medical chemist field, and the three of you come together and develop something. In Europe, it's called the bed to bench to bed approach, bed with where you sleep in, you know. In Canada, it's very hard to do, and yeah, you see the collaborators we have is still in you know, it's what it is. Technically, you know, my guys, if you wanted to come to me, we are hardcore structural biology people and biophysicists. We have lots of machines. Again, this CRC system was very generous to me by getting money over the years. I think I think we got $12 million worth of equipment from Western Economic Diversification, from CFI. We have a huge machine park, and we love to collaborate with people. But at the end of the day, it always starts with an intelligent question, and therefore technically we would require a strong med school. So we work with the guys in Cologne or in France. Patrick Mélin is the big boss at the French Cancer Research Council. And we need people who synthesize compounds, therefore we work with Atomwise. This kind of integrated approach, you know, when I was a grad student, when I did my PhD at Max Planck with Bob Huber, who got a Nobel Prize in '88, you know, we were trained to be excellent crystallographers, and I got a job, and I moved to Basel because I was obviously not that bad of a crystallographer. Today, nobody cares. Today, everyone is, and I get the point, people want to have an interesting question and you to solve the problem. And students and postdocs and so on working with me are always encouraged to learn as much as possible of all these techniques I showed you to find an answer to an interesting question. You know, these days rarely you get a job because you just can solve crystal structures. This is past. People are attracted, and I am in so many search committees and advisory boards for synchrotrons and you name it, that, you know, it's this integrated, you know, come together kind of thing. Dean Martin says again, is there a potential for Netrin to be used as a diagnostic marker? Yes, it's a big Netrin 1, and we published a paper on Netrin 4. They are biomarkers. Yes, they are. Okay, Jason. Okay. Oh me, is several. Okay, no. No further questions. Thank you.
All right. Thank you so much, Jörg. Lots of thank yous to you in the chat as well. We really thank you very much for fitting us into your very, very busy schedule and agreeing to do this on a rather short notice. That's really appreciated. So okay. I'm going to now move to advertising, I guess, our next speaker. So our next speaker is going to be an alumnus from U of M, David Owasi, on May 15th, Friday, same time, who's going to talk about emotional intelligence. And so this would be particularly of interest to students because he's going to tell us about how essential this is, and particularly for the job market. So we hope to see you again next Friday. Thank you everyone for your participation. So we look forward to seeing you again. I guess this talk filled up rather quickly, Jörg, so we're gonna be forced to move to another platform next week to accommodate a lot more participants. So thank you for your patience. And so we hope to see you next Friday. And until then, please keep well and have a wonderful weekend. That ends the session. Bye for now.